This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 249, recorded on August 19th, 2021. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. Nice to be on.
Nice to see you again. Welcome back in this August of 2021. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. Happy nearly end of August.
Yeah, isn't that fast?
How did that happen?
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Michael, you haven't had any huge storms lately, have you?
No, it just rained for 2 solid weeks. So the grass is very green and growing like weeds.
How about mushrooms?
Mushrooms came up. I have to send you a couple of pictures that took.
Please do.
Elio, I keep getting ads for mushroom coffee. Have
mushroom
you ever heard
coffee?
Of that? Yeah.
What the hell is that?
You see it too, Michele? Yeah.
Yeah, on the Pod Save the World podcast.
They have ads for mushroom coffee?
Yep, they sure do.
I got one on Instagram, and it's like, it's gentle. It doesn't jolt you awake. And I said, well, that's good to be jolted awake.
That's
Not why
what you drink coffee for.
Yeah.
Ay.
It's funny. Mushroom coffee. I guess it's made from mushrooms. All right. Well, that's, it's a microbial product. It's good. Here on TWiM, we love microbial products. And so we are back for your education in all things microbial. And today we have, of course, 2 papers for you. And we're going to start with a snippet from Michael.
All right. So this snippet is entitled Temporal Shifts in Antibiotic Resistance Elements Govern Phage-pathogen Conflicts. And this paper appeared in Science in the 30 July issue. And it's from Legault, Hayes, Angermeyer, McKittrick, Johura, Sultana, Ahmed, Alam, and Seed. And they are from the Department of Plant and Microbial Biology at the University of California at Berkeley, as well as the International Center for Diarrheal Disease Research in Bangladesh, as well as from the Chan Zuckerberg Biohub in San Francisco. Now, this snippet will offer us again an opportunity as we wind up our summer holiday here in the Northern Hemisphere to ponder the wonder of nature and to consider how something like time-dependent shifts in antibiotic resistance elements can impact, govern, perturb these phage pathogen conflicts. In fact, many of us fail to appreciate that in nature, bacteriophage predation has been estimated to account for up to 40% of all bacterial mortality. And in fact, in Bangladesh, where the microbe that we're going to be discussing today, Vibrio cholera, is endemic, and lytic phage predation is implicated in modifying the duration and severity of cholera outbreaks. So we all appreciate, but often in the isolation of our model systems that we work with in our lab, we forget or neglect to consider that bacteria are in this constant dynamic struggle with an abundant and diverse array of bacteriophage. And I always tell the students that come into class that, the seawater that they're surfing in out at Folly Beach can actually have 2.5 million phage per mil of seawater. So the microbes really need to hold on to an extensive arsenal of defensive systems in order to live, if you will. These so-called anti-phage systems, now we know from our genomic analysis, cluster within something that has been generically described as the mobilome. And the mobilome, as you can well imagine from anything with ome, is a set of genes that move about. And mobilomes are present in eukaryotes, bacteria, and viruses as well. Viruses can actually hop between other viruses. And I guess the best example that many of you would be familiar with is, of course, AAV and adenovirus. AAV needs adenovirus in order to be infectious. And so it's, if you will, part of this mobilome of viruses. So let's go back to thinking about how to introduce this concept. We know in bacteria, mobilomes, or we know them as plasmids, or in the last TWiM, we learned about the Borg, and resistance is really the assembly of these gene products that live in this genetic element, that really through this mobile genetic element flux is really driving this predator-prey behavior. And even though the editors from Science introduced this paper in their editor summary as Back to the Future, I'm not going to use any analogies to flux capacitors or anything like that. What piqued my interest, and it's why it's on the top of things to think about this summer, is we lack biologically relevant systems to study ongoing phage-host interactions. And this is apparent, particularly apparent when you begin to consider this from a clinical perspective. Now, what these authors did is through long-term sampling, and long-term means they looked at the cholera-infected patients, the bacteria, as well as the phage, from 1987 through 2019. So we have about 30 plus years of cholera bacteria as well as the phage from Bangladesh, and they revealed the persistence of 3 lytic phages, ICP1.
And Michael, if I could just interrupt for
Sure.
a moment, I would like to just give a huge amount of credit to Professor Shah Farooq, who's a professor at the School of Environment and Life Sciences at the Independent University in Bangladesh. He has really been the leader in collecting these samples over, as you said, a 30-year period, and has been a really key collaborator with many of the major cholera groups in the U.S., but globally. So Rita Caldwell, John Mekalanos, Andy Camilli, Kim Seed, they have all been able to do really sophisticated genetic and bioinformatic analysis, as we'll see in this paper. But thanks to this huge collection of samples over decades.
And that really is the key, Michele, is that it's only through these samples that we are going to begin to gain a limited perspective of the phage-host coevolution, if you will, the yin and yang that we are witnessing today with another virus in its host, namely SARS-CoV-2 in the human race. Yet, fortunately for us, bacterial genetics is much simpler than human genetics. But the bacteria, what we're going to learn at the end, if you will, have their own immune system that is effectively able to interact with the phage and effectively throw curveballs at it to try to maintain this yin and yang approach, or as the authors tease out in their discussion, it really is an arms race between the bacterium and the phage, each one developing a different weapon in order to combat things. Now, the way they go about this is they offer us insight that only can be developed, as you said, Michele, from this longitudinal sampling of this pandemic-causing microbe. And because it causes a pandemic, we really see this in a large number of people, that we can get a large number of samples that we normally wouldn't see if we were doing this in a lab, because you just didn't have the real estate in order to perform these sorts of experiments.
Yeah, so these are clinical samples, and then they also have samples from the waters, phage from the waters, from clinical samples. It's an amazing resource for the whole Vibrio community.
In brief, the authors performed what they term a time shift assay, which is beautifully illustrated in their first figure. and as well as cited in the references. So I encourage you, if you have an opportunity, go back and look at those papers, because it really gives you an understanding of the work that goes in to looking at it. Now, what they did is they did one of Rich Condit's favorite things. They did plaque assays. And in their first figure, they show us a beautiful plaque assay. And you see the plaque assays, and and this is specifically figure 1B, and it shows they challenge Vibrio cholera isolates with phage recovered from patient stool samples from the relative past, the present, and the future. Now you're going to say to me, how can you have phage from the future? And the answer is because these are longitudinal samples. So they can take a 2019 phage and plaque it on, say, a 1990 bacterium. And as they described in their paper, a pattern emerged in which Vibrio cholera isolates were uniformly susceptible to the contemporary phage, but it restricted the one lytic phage, ICP1, from temporally mismatched patients. So phage from the past couldn't plaque it, and phage from the future couldn't pack it. And you just follow their figure down, and it's a perfectly diagonal line as to what can plaque and what cannot plaque. Now, here's the cool bit. They walk us through how they uncovered the mechanism of phage resistance in these toxigenic Vibrios, and thereby offer us how only the contemporary phages could still plaque the host. And this is a really simple assay. It just simply says, can you lyse the host? But the ones from the past and the future failed. Enter the power of this, if you will, comparative genomics with the time shift results. What they found was resistance, or the inability to plaque, was associated with fluctuations in what they term an ICE element. And ICE just simply stands for an integrative and conjugative element. And so this is nothing more than what we learned in simple microbial genetics. It's like an fertility plasmid. It can integrate, it's self-mobilizable, it can do its own excision, and then it can literally have sex with another host and transfer the information. And this was principally associated with one of these ICE elements that they abbreviate SXT, which stands for an antibiotic suite marker. And it happens to be sulfamethoxazole trimethoprim. And it's on this cluster. And I put a link in the show notes from a Frontiers in Microbiology paper on this SXT integrative conjugation, or ICE element. And the title of that paper is A Novel Trapdoor Strategy for Mobile Element Escape. And that really will give you all the background you need to know as to how these microbes are effectively having this arms race. What we do know about this SXT integrative or ICE element was they simply harbor antibiotic resistance determinants. But when we look more carefully at this very large segment of DNA, we find that there are hotspots. And these hotspots are now appreciated to be associated with the genomic traits of the 7th pandemic strain of Vibrio cholera. And these ICE elements, or SXT ICE elements, are infamous for conferring multi-drug resistance. And they also bring around cargo genes that confer a broad suite of adaptive traits to their bacterial host. And their observations found that this SXT ICE element from these clinical isolates of Vibrio and other gamma proteobacter carry signature sequences for these defense systems. And they are things that you probably remember, like restriction modification systems. And, anyone who does molecular biology takes advantage of restriction enzymes, which are a component of the restriction modification system. and then something else called the Brex system. And Brex simply stands for bacteria phage exclusion. And this was described originally in Bacillus, specifically Bacillus cereus, and it's a series of 6 genes that actually confer the ability to exclude phage. And what we'll learn here in this paper is that the phage actually makes a protein that can counteract Brex. So it takes out the Brex system. And it's really pretty interesting. Now, the power of this manuscript is they have 2,600 toxigenic Vibrio cholera genomes. And they revealed that there was this temporal dynamo of this ice element. And they also appreciated that there was an unexpected persistence of strains that lacked this element. They were actually deleted for this SXT ice element. And the authors then went further to characterize the 3 dominant SXT ice elements in Vibrio cholera, identifying the defense systems as actually having this ability to effectively get around the Brex system. They offer us a mechanistic understanding of what happened. And what they go into is they talk about fitness. And normally they talk about the burden of carrying this extra genome around, if you will, this extra bit. And it's a large segment of DNA that these are carrying. So it's truly a fitness penalty. It's the equivalent of my poor sister-in-law who commented that she overpacked on her visit to my house as my brother muscled her suitcase into the car. And, so you sort of want to leave behind things so that your luggage is not as heavy. So they argue that the way the phage escape the defense mechanisms of the host is either through 2 distinct mechanisms: the epigenetic escape from restriction modification, or through this novel anti-Brex inhibitor protein that they identified, ORBA. Now, they did a lot of work over this 34-month surveillance period. So why does this all matter in the great scheme of things, and why we need to consider this? Well, there are identical sulf-methoxazole trimethoprim ice elements are found in multiple taxa, with certain antibiotics stimulating the high frequency of transfer. Remember, antibiotics are selection pressure, prompting the authors to ask whether infection by phages can overcome this antibiotic selection mediated defense, or whether or not conjugation is actually driving the concurrent dissemination of phage. And as you might guess, it's antibiotic resistance. But from the phage and microbes perspective, it really is indeed these adaptations that are the yin and yang going on between the microbe and the phage that are allowing the phage to effectively overcome. Now, the authors put this into perspective by offering us that the phage, specifically the lytic phage ICP1's adaptations to overcome the contemporary ice elements are driven both by changes in its capacity to express the anti-ORB protein and the acquisition of epigenetic modifications, allowing for the infection of these other ice element containing Vibrio cholera. Bottom lining this for your consideration is that these ice elements determine phage resistance in clinical isolates of Vibrio, with the selection of phage encoded counter adaptations that may be lost as these antibiotic selection factors fluctuate in dominance due to the selection pressure, leading them to offer as a conclusion, we have a heterogeneous pool of these SXT ice elements constitute, if you will, a pan immune system that is able to restrict all phages and mobile genetic elements that they actually tested, and that a SXT vibrio SXT deletion vibrio may confer a population-wide benefit to vibrio by limiting epigenetic escape, leading, as the authors say, to subsequent bottlenecks that maintain the anti-phage benefits afforded by these ice elements. Reducing this all to a soundbite, phage and antibiotic resistance together on a single mobile genetic element whose transfer is stimulated by phage infection. This paper goes into so much more detail, but it really can be simply stated that it's a yin and yang. The phage are reacting to the host ice element is doing its thing by mixing in the environment, and they beautifully show this through their very detailed and elaborate scheme that they develop in their paper.
Michael, this is really all about Brexit, isn't it?
It is. It is about Brexit.
Punny.
This figure 3, by the way, I love the images they've drawn. There's one figure where they have diagrams of T2, T4, T5, 6, and 7. It's just gorgeous because you see the little
which are all phage. Those are each
all phages, and the short tail of T7, the longer one of T5. They're just so cute. I love it.
It was really a hard paper to do justice to in the short snippet, but I hope I didn't butcher it too badly for you folks. But it's really something that you should take a look at.
We got the bottom line. I think
if I could just amplify, I agree. And Michael, you pointed out that the phage are driving conjugation of this mobile element, but also antibiotic resistant or antibiotic treatment triggers mobilization. And so clinically, we of course rely on antibiotics, but there's also now interest in using phage therapy as a new approach to taking care of infections. But now we find we've got one genetic element that can conjugate and move between species and confer antibiotic resistance and phage resistance. Yeah. So it's amazing what is happening out there in evolution, and it has direct impact on our strategies for treating human infections.
Yet, at the end of the day, phage still account for 40% of bacterial death.
Yep.
Yeah. So, it's not only just beautiful biology and microbiology, but it's got implications for human health.
Well, I think, we did a paper recently on phage therapy. And I think if you bookend your thinking about phage therapy and contrast it against this paper, it will really tell you the limitations that are going to need to be overcome by the pharmaceutical industry in order to understand whether or not phage therapy will ever be a viable solution. Because the bacteria certainly have figured out the yin and yang of getting around phage.
Right. So we'll have to use cocktails, much as we use cocktails to treat Mycobacterium tuberculosis, right? We've got to come at it with multiple. Yep. Really beautiful work from Kim Seed's group at Berkeley.
Very nice. Another Berkeley. Last time we had, remember, another Berkeley group with the sampling of the sludge out there. Remember that?
Oh, yeah.
Yes.
I forgot the name, but another Berkeley professor. Thank you, Michael. All right. Now we have from Elio something very close to home.
Yes. The article is from MBio. It reads, intracellular localization of the proteins encoded by some type 2 toxin-antitoxins systems in Escherichia coli. And it's by Alexander Magel, Tommy Safran, and Hannah Engelberg-Kulka, who happens to be my cousin. So this is in the family. Okay, I don't think we talked much about toxin-antitoxin systems on this
I don't think so either.
site, have we?
No, I
No.
don't think so.
Okay, here is one. It's a story that is really has fascination and puzzlement both, and this is the reason why. Widespread in bacteria is a system called toxin-antitoxin. Two proteins, usually two proteins, sometimes not, which are expressed together. One of them is a toxin, that is, it causes damage to the bacteria, and the other is an antitoxin, which neutralizes that, an antidote, if you wish. Now, this has been known for about 30 years now. It started out with the discovery by Yarmolinsky that plasmids encode for such proteins, and that if the antitoxin is not working, the toxin will be working and kill the bacteria that carry the plasmid. So later on, in the lab of Engelberg indeed, she and collaborators discovered that such systems exist not just in plasmids but in the chromosome. And it turned out that this is a huge topic because most bacteria carry more than one of these systems. The numbers are so staggering that in some tubercle bacilli, I think bovine strains, there are something like 50 different toxin-antitoxin systems. Can you imagine that? I mean, and this has escaped the discovery for quite a while. So anyhow, the subject is not without its controversy, and it's been argued for some time about the meaning of all this. And this is not quite what we're talking about here. Here, the lab of Engelberg at the Hebrew University in Jerusalem asked, where are these proteins located inside the cell? What is the intracellular location of these proteins? And this is something new and different, but of course, very popular these days because we live in an imaging world. We're really interested in seeing where things are inside of cells. And due to mainly the availability of certain fluorescent proteins like M-cherry, which can be cloned into the genes in question, we can find out where on Earth, where inside the cell are these proteins located. And this essentially is what the paper is. It's a surprising paper. It's the first, I'm sure, of a large collection. And if it's true to style, it will be controversial. But so far, we have a good story, and the story is pretty straight. So first of all, it turns out that cloning M-cherry into these proteins does not affect their ability. They're very careful to measure that to find out that in fact not much has happened to the system by just introducing M-cherry. That's very nicely done because it's a crucial point in trying to understand where we're going with this. So the toxin-antitoxin systems are divided into types. There's type 1, type 2, type 3, and there's more. There's 5 or 6, in fact, total. And they're all a little bit different. Type 2 is the classical one, and it started out, as I say, in the discovery in Engelberg-Kulka's lab of proteins, two proteins, which are manifest upon starvation. When you starve for amino acids, you find that these two proteins come up. And when she saw this on a gel, Engelberg Kulka asked the question in Hebrew, "Mazeh." Mazeh means what is this? Mazeh became Maz-E in English, which turns out to be antitoxin. I'm sorry, the toxin, and Maz E and Maz F, because F follows E. So E is the antitoxin. No, E is the toxin. F is the antitoxin. So there is mass E and mass F in English.
And
Anyhow,
they're really toxic. It will kill bacteria dead if you don't have the antidote.
That's right. That's right. So the question then becomes, so there's a lot to this, and I won't go into it because, as I say, it has a history of its own. There's quite a literature about this, and you're welcome to go find it. It's
and
not,
quite a
not
dogma associated with how these
that's
work.
Right. That's right. Actually, I think that the reason for the controversy is that people have approached it in a somewhat dogmatic way. Anyhow, the question then is, where are these proteins located? And the answer is, if you look at with M-Cherry labeled systems, you find that they're different. And this lab studied not one, not two, but 4 different kinds of toxin-antitoxin systems. They're known as MAS-EF, CHPB, M9SRA, etc., RNLAB. Not all of them are well studied, but they went to the trouble of making these constructs with M-Cherry for at least 4 different toxin-antitoxin systems.
And if I could point out, in each case, the toxin targets mRNA and degrades mRNA.
That's right. That's right. Thank you. So what's the story? The story is that all 4 are different. Not one is alike. Let's take MAS-F. In MAS-F, if you just look at the toxin, it localizes at the poles. If you add the antitoxin is dispersed in the cell. And if you have both toxin and antitoxin, they're in the middle of the cell. Now that's surprising because the toxin is normally by itself just in the poles. So here's, without even going into what the other 3 systems do, this is really weird. The toxin is at the poles when it's by itself. When it's with the antitoxin, it moves to the middle of the cell. Why? They don't know. They're very blunt about telling us that they don't know the answer.
But it's quite striking and in vivid color before our eyes.
Vivid color.
That's right.
Yeah.
It's very striking, and it's statistically very solid because they look at a lot of cells and they have optical methods for doing this and computer.
So it's quantitative data, hundreds of cells.
So let's say that this looks real, and what does it mean? It's up to you, dear listeners, to figure this out because nobody knows at this point. Why would a protein, which is normally located in the poles, move to the middle of the cell? Well, the middle of the cell is where this future septum is. So maybe it has something to do with cell division. Maybe it has something to do with something like that.
Sorry, Elio, what moves to the center? Both or just one?
Both.
Both move. If you just, let's say you just put the antitoxin in alone, does it move also to the center?
No. It's diffuse.
It needs to have both. And of course, you can't have the toxin alone because it'll kill the cell.
So we're discovering not only some answer to the question of how these systems work, but something brand new in cytology, microbial cytology. This is really new. The movement of a protein from one part of the cell to another part of the cell by help of another protein. What's this all about?
And what's so striking is that the assumption had been made when the antitoxin is bound to the toxin, it just inhibits its enzymatic activity. So that's how it shuts it off. But it does more than that. It does more completely changing its localization. So this is really disrupts the dogma in the field and opens up a new, really exciting area of biology.
And this is why this paper could be controversial because, the dogma before was that toxin and antitoxin get together and it takes out the toxin activity. But this
and that's still true.
That is still true. But the localization really gets to the mechanism of how the toxin is inactivating the microbe. And I think the other thing that we need to think about is that given the volume of a bacterial cell is so small, the normal expression and understanding the stoichiometry of how much toxin is produced in relation to the concentration of antitoxin becomes important to understand. And that's what a lot of the literature plays around with is ratios and stoichiometric relationships, which also drives some of the controversy.
By the way, when they look at the other three systems, I told you they looked at four systems, they're all different. Each one is different. My God.
But in general, 3 of the 4 look similar in this shift to the center of the cell.
There's a shift to the 4, that's right. But the details are different in each one of them.
Right.
So we have new microbial cytology to deal with. This is really quite fascinating, I think.
And it points out like genetics are a great way to study things, biochemistry, another great way. But unless you look in the context of the whole cell, you might be missing a layer of regulation or coordination.
This reminds me of the work of the Polianos, right, who are very much into cell biology. So, Elio, there's this cytoskeletal structure in bacteria. Does this localization have anything to do with that?
They wonder. They mentioned that in their paper, but they have no data to support that.
Yeah.
They also know that the antitoxin triggers degradation, proteolysis of the toxin through a protease called ClpP, and so they don't know whether position at the pole versus in the center changes accessibility to this protease. It's another hypothesis that they can now test.
Yeah.
Did they look at the protease localization here?
Not yet. No. This is really the birth announcement.
Interesting. But maybe, Elio, for the listeners who haven't heard of this, what would be the function of having a toxin-antitoxin system in the life of the bacterium?
Well, they're set off by starvation, by antibiotics, by UV radiation. In other words, when the bacteria are stressed, they express the system.
Okay.
And so that's the connection.
They also mentioned for one of the toxin-antitoxin systems, the specificity of the RNase is to a particular 3 nucleotide sequence, and that particular sequence is absent from mRNAs that are important for biofilm formation. So one idea is that when a cell is stressed and they want to shift from, let's say, planktonic growth to biofilm growth, this toxin-antitoxin could clear out the cell of the planktonic RNAs and favor accumulation of the biofilm. So it could be part of the developmental switch, rapid, efficient developmental switches. And of course, recycle all the nucleotides that are in RNAs that we don't need anymore. We want to switch and make a different set of RNAs.
Which is really important in biofilms.
Sure. Yeah. Are these toxin-antitoxins in widespread species of bacteria?
Yes.
Yes? Not just E. coli and Pseudomonas.
Oh, no. They're in TB. So, all kinds of bugs have them. In fact, the numbers are staggering. They say in some systems, like in TB, in bovine TB, there are about 50 of these systems in one single cell. Go figure.
And the expression of them, some of them is linked to accumulation of a very widespread alarmone, PPGPP, that turns on the stringent response. So it's clear that these toxin-antitoxin systems are plugged into the fundamental physiology of cells. So a lot left to learn.
Yeah, it really is an eye-opener in the sense that here we think we live in a time when we understand at least E. coli, if not some other bugs. It turns out there are new things that we have no inkling about.
Even though it's a very simple two gene system.
Right.
So the person driving this work, Alexander Mager, is a PhD student in Engelberg Kolka's lab. He's originally from Russia, and he did studies at the State Medical Academy and then went on to work as a doctor of clinical laboratory medicine in the regional hospital in Kazan, Tatarstan, Russia. After doing that for a while, or while he was there, he was in charge of the daily routine laboratory tests and quality control particularly in the perinatal center of the hospital, and also was responsible for introducing new clinical tests and new analytical methods in the lab. But he then moved to Israel and was fortunate to join the Engelberg Coca lab and is working there on his PhD, and hopes that this project will also lead to a better understanding of new antibiotic strategies. He was surprised, as we were, that his work really challenged the central dogma in the field, that it's simply the physical interaction. So he's excited about this discovery that it changes the localization. And when Alexander is not doing these experiments, he's a musician. He plays guitar and harmonica in a couple local bands, and he's often invited as a session blues and jazz harmonica player in the studio, recording studios. His wife has a Russian bookstore in the center of Jerusalem that has become a cultural center that hosts lectures, exhibitions, and live music concerts. The bookstore is called Babel Jerusalem, and it's named after a famous Jewish writer, Isaac Babel. So I'll put a link to that bookstore in the show notes. Alexander does have advice for junior colleagues, and he says, investigate a fascinating subject in a well-experienced laboratory, which he certainly does, and he is very grateful for the opportunity to work with Hannah Engelberg-Koka and her colleagues.
Hey, Leo, do you ever communicate with your cousin?
Yes, we do. We communicate once in a while. I have only 2 cousins in the world. This is one of them. The other is her brother. So we communicate a fair amount, yeah. We love each other dearly, so...
So did you have any role in her becoming a microbiologist?
Actually, no.
Or vice versa. Vice versa.
What's that?
Or maybe she inspired you to become a microbiologist.
Neither. I was living in Ecuador when my parents and I decided to take a trip to Israel, and I saw Hannah for the second time in my life, essentially. I'd seen her when she was very young. And I said, what do you do? She says, I'm a microbiologist. Guess what? I'm a microbiologist too.
Oh, wow.
We had no clue. We had not communicated for years and years.
Wow.
Living in 2 different parts of the world. And when we finally met, we realized we were both in the same field. So she's been doing extremely good work. And so I'm very proud of her.
Very nice. That's a cool story. Independent evolution.
That's right.
Yeah.
Convergent evolution towards microbiology.
And one of the things that we should point out is this is only a 3-author paper.
Yes.
I mean, the amount of work in here is incredible.
Yep.
And this is only a 3-author paper.
Yeah, they don't acknowledge, but they must have had a lot of help with regard to this. They acknowledge the help from the cytologist.
Yes.
And also, we didn't have time to go into it, but they were very careful to do controls to make sure that these fusion proteins were not disrupting the behavior of the toxin and the antitoxin. And a lot of really careful, rigorous quantitative work.
Yes.
All right. Thank you, Elio. Thank you, Michele.
Yeah.
Our pleasure.
Let me read one email we have, which is somewhat relevant to today. This is from Vivek, who writes, hope you're doing well. I'm sure you don't remember me, but we met during one of the coffee breaks at ASM San Francisco in 2019 and talked science. Boy, that was the last time ASM ever met, right?
Yeah.
Yes.
Cheapers.
This is the last time we were all together.
Yeah, I do remember that. That's right. I also talked about how much I love TWiM and TWiV team and program. Here's the pick for us at 2019 was when the world was sort of normal. I have a picture with Vivek.
Great.
I'm writing to share my groups and Arkin's group at Berkeley's work on phage resistance landscape at a scale that was not done previously. Thought this might be a good paper to highlight if you have a spot to talk about phages. He provides a link to this paper, which is in PLOS Biology. Essentially, we used classical textbook phages from the bacteriophage book edited by the late Professor Rich Callender against loss of function and gain of function libraries of 2 strains of E. coli and uncovered diverse overlapping and distinct modes of resistance. We found many dozens of host factors, even for the well-studied phages, specifically for N4 phage. Since this work, we have extended these studies to gain mechanistic detail on bacterial interaction with diverse predators, antibiotics, metals, and antimicrobial peptides, and have been using the technologies for other applications. Maybe the general audience will appreciate that we are working on improving our understanding around how we are characterizing viruses that are friendly to us. Finally, this paper is very special for me. I got the opportunity to collaborate with Professor Callender. He passed away on the same day this paper was published in PLOS Biology in 2020. Really appreciated his excitement about my work, his guidance, and friendship. Phage research community has lost a giant.
Yeah.
Thanks to you and the TWiM and TWiV team for making science communication authentic and exciting. Best regards, Vivek, who is a scientist at the Lawrence Berkeley National Laboratory in Berkeley, California. So thanks for writing, Vivek. Long time ago. Boy, time passes when you have a pandemic, doesn't it?
It does indeed.
I thought his letter and also our papers today really highlighted, although there's been just a ton of excitement around CRISPR-Cas as a predator-prey system in microbes, there's still a lot out there going on that we haven't studied yet. And it's going to be rich and really could inform therapeutics.
It used to be that 40% of the genome of E. coli was not understood. We didn't know what's in there. So I don't know what the number is now. Maybe it's smaller, but it's still a significant number of genes with really no clues to what they do. And believe me, they're all important. Otherwise, they wouldn't be there.
That's right.
And of course, those predator-prey relationships between bacteriophage and bacteria have been around a whole lot longer than the rest of us. So it's sure to be a rich area of research.
So, Elio, our chromosomal DNA, less than 2% codes for protein, right?
What?
Less than 2%. So what do you think about the rest, Elio? Do you think that has some role, or is there some junk in there?
I think a lot of it has some role.
So a lot of it's repetitive elements, introns. A lot of it's integrated dead retroviruses.
Like debris.
Which I think there's a lot of room, and you can carry things for years. I mean, some of it may be useful, but there's a lot that probably isn't. And
sort of like the trunk of your car.
Yeah, a place funny. You can put new things without disturbing the rest of the function.
Yeah.
Michael, when I get a new car, I take all the stuff, put it in a box, put it in the garage, and then the new car remains empty until it accumulates its own stuff.
And then you have another box in the garage.
And then you have another box, and it's like the genome of my car. It's funny.
Those are called plasmids.
Plasmids, yes.
Yeah, the box is a plasmid.
It's the mobilome, right?
It
is the mobilome. Great analogies. All right. That is TWiM 249. You can find the show notes at microbe.tv/TWiM. If you want to send us a question or a comment, we'd love to hear from you. My TWiM, TWiM, at microbe.tv. And if you like what we do, consider supporting us. Give us some contribution. Microbe.tv/contribute. Michele Swanson's at the University of Michigan in Ann Arbor. Thank you, Michele.
Thank you. What a pleasure and privilege to talk about this really cool microbiology that has such deep implications.
Elio Schaechter is at Small Things Considered. Thanks, Elio.
My pleasure. I really enjoyed it.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.